There are three mechanisms of gene transfer in bacteria: conjugation, transformation, and transduction. Conjugation is a direct physical interaction. It's oftentimes referred to as bacterial sex for that reason. And you can see in this very simplified diagram, there's a physical opening created between the two cells, and genes are going to be able to transfer between that opening. Transformation is when DNA is released from a dead cell, and then that DNA in the environment is picked up by another cell, which can then incorporate that DNA. So a dead cell, a cell dies, releases its DNA, and a different cell picks up that DNA. And then the last one is transduction. This is when a virus will accidentally transfer genetic information from one bacterium to another. And this will actually negatively impact the virus, but can be a benefit to the bacteria. So conjugation, as I mentioned, is oftentimes referred to as bacterial sex, and that's because it's going to involve a physical interaction. So it happens between an F positive and an F negative cell. This refers to having the F plasmid, or remember the fertility plasmid. So only a cell that has the F plasmid is going to be able to initiate this. A cell that has the F plasmid will make a structure called a sex pilus. And the sex pilus will move from the F positive cell to an F negative cell and attach to it. So the F positive cell sends out this structure to the F negative cell and attaches to it. Once that physical connection is made, the two cells get moved closer together and create this opening between the two cells. So there's the opening between the two cells. There's the bacterial chromosomes, but it's really the F plasmid that's important here. This F plasmid will get broken open, so it'll go from being a circle to a line, and then that line can snake across the connection between the cells. So we'll end up with two cells that have their original chromosome, but then each one has a single strand of the F plasmid, and then those cells will copy that and make the second strand. And so you go from having an F positive and an F negative to having an F positive and another F positive cell. So again, the strand of the F plasmid is cut, and that allows it to come over to this side, and then they each end up with one strand of the F plasmid, so that has to get copied to make it double-stranded, and then now they're both F positive. It's also important to note that while the F plasmid is important for making this sex pilus structure, the F plasmid can also carry other genes. So for example, the F plasmid can carry resistance genes. So conjugation becomes one of the ways that resistance will spread between bacteria. The second method of gene transfer is transformation. And so remember, this starts with a cell dying somewhere. So when a cell dies, so here's my cell, it's got its DNA. When it dies, it breaks open. So the membrane breaks open, there's all these pieces of membrane, and then the DNA also gets broken into pieces. So there's also all these pieces of DNA floating around. And this just becomes part of the environment. So in any environment, there's lots of pieces of broken down DNA, broken down RNA, broken down proteins, broken down membrane, all floating around. So then you have a living cell that's in that environment. So this is the environment full of all these pieces of DNA and RNA and proteins. And some cells, some bacterial cells, are what's called competent. So this is a competent cell. What that means is that it has this. It has a receptor for pieces of DNA. So because this is a competent cell, it has this receptor, and it can pick up a piece of DNA in the environment. So these receptors will take that piece of DNA and bring a strand of it into the cell. So here's the original chromosome, and then it's going to bring in that little piece of foreign DNA. Once that little piece of foreign DNA is in there, there's the possibility, if it matches, so it has to be homologous. Remember that term homologous, similar. If it's similar enough, it will get switched out, just kind of like crossover. It's a really similar mechanism to crossover that we learned about in eukaryotes. But if it does match, what will happen is that little blue piece will get exchanged with a little red piece. So a little red piece will get cut out and become trash, and the blue foreign piece will get integrated into the bacterial chromosome. In the image here in your textbook, it's following this gene, which is for tetracycline resistance. And so what's happened is now that this is part of this chromosome, this would be resistant to tetracycline. So a bacterial cell can die that's resistant. It gives off its genetic material, it floats into the environment, and if there are cells that are competent out there, then they're going to be able to take that foreign piece of DNA and change their own genetic makeup. So again, this is a way that resistance can spread among bacteria. Now, I just want to remind you that this process is only going to happen with DNA that has some matching. It doesn't have to be necessarily the exact same species or the exact same strain of bacteria, but it can't be a human gene. Like bacteria are not going to pick up and become transformed by human genes. They can't do that. It has to be close enough genetic match in order for transformation to occur. Okay, the third method of gene transfer is transduction. And transduction is going to occur through a virus. So we've looked at the viral reproductive cycle. So remember that when a virus comes into the bacteria, so here's the virus entering the bacteria, it's going to want to take it over and turn it into a host reproductive or turn it into a virus-making machine. And part of doing that, and I didn't necessarily emphasize this when we're talking about viruses before, but part of doing that in the case of the phage is to take the host DNA and just destroy it. Just cut it into lots of little pieces. Because if I break up the host DNA, the host can't do anything with its own DNA, right? So this is a way to take over the host cell. So if I look at my infected virus, or sorry, my infected cell, it's got lots and lots of pieces of its own DNA. So it's been broken up into lots of little pieces. And then it's also got the viral DNA. And remember, the goal for the virus is to synthesize lots more copies of the viral DNA and to, of course, make viral proteins and wrap it around that viral DNA. But remember that in around all of these pieces of viral DNA, there's also all these pieces of bacterial DNA, because it got broken up, it's floating around in the cytosol. And so sometimes what accidentally happens is that during viral assembly, the capsid gets wrapped around bacterial DNA. So again, this is bacterial DNA, not viral DNA, that got packed inside of the capsid. Is this good for the virus? No, this is bad for the virus because this is not going to make new viral particles. It doesn't have the viral DNA. Okay, but because it has the capsid, it can still go out and enter another bacterial cell. So this is the infected cell. Remember that the virus is going to burst out of that infected cell and kill the infected cell. And out comes all of these viral particles. But if this viral particle goes to a new cell, so here's the new cell, here's that viral particle landing there, and I'm making it much bigger just for simplicity. It's going to send that into this new bacteria, and there's its bacterial DNA. But because this isn't the viral DNA, this isn't going to turn this into a viral machine. Instead, what can happen is, just like with transformation, if there's some matching between these two things, then this will get put inside of the bacterial DNA and a piece will come out. And so now you have a new gene in the bacterial chromosome and no viral infection. Because again, it was a piece of bacterial DNA, not viral DNA. And it brought in a new gene, but it did not cause a viral infection. And so again, this is a way that traits can be passed from one bacteria to another. I just want to one more time say the virus is accidentally doing this. It's not in the interest of the virus, but it is one way that genes can be transferred among bacteria. So all of these gene transfer methods are what's called horizontal gene transfer. And this is part of horizontal evolution. So normal gene transfer, what we traditionally think of gene transfer is that you have a parent and they pass their genes to their offspring. And through that process, right, this is also how evolution occurs. So one species will become another species as changes happen between parent and offspring. And this is what's called vertical evolution or vertical gene transfer. But what bacteria can do is they can exchange genetic material across, so not parent to offspring. And because it's not this linear downward route, it's called horizontal, not vertical. And it's interesting that to realize that in bacteria, horizontal gene transfer accounts for a large majority or a large amount of their genome, not a majority, but a large amount of their genome. So when they look at, for example, some different species of bacteria like E. coli, almost 20% of their genes were acquired through horizontal transfer. So that means that horizontal transfer is a major player in determining traits and genetic material of bacteria. And it's super important to realize this in a medical context as well, because if 20% of the genetic material of bacteria is coming through this horizontal gene transfer, then the traits that we don't like in bacteria, like antibiotic resistance and pathogenicity, are also being acquired through these mechanisms. And so it's something that we have to be aware of, especially when we're trying to control antibiotic resistance in places like hospitals. So that is going to be a challenge for humans as we go into the next 50 years as more and more of our antibiotics stop working.